Hello and welcome to Bio40 Pre-Lecture 21. Today we finish our discussion on translation. Similar to how we describe transcription, translation can be divided into 3 steps: initiation, elongation, and termination, though the details of each step are very different. In translation, initiation begins with the start codon, AUG, beginning the polypeptide chain with methionine. Chain elongation is mediated by molecular machines called ribosomes, which proceed along the mRNA in the 5' to 3' direction, extending the polypeptide chain with the amino acid corresponding to the proper codons. It does so at a rate of 2 to 15 amino acids per second. This continues until the ribosome reaches a stop codon, which does not correspond to any amino acid. At this point, the ribosome falls away, and the completed polypeptide chain is released. Let's talk about the ribosome, the key player in all of this. Ribosomes are complex molecular machines composed of both protein and RNA. The ribosomal RNAs are called rRNAs. The ribosome has many functions. It recognizes the mRNA, initiates translation, stabilizes interactions between tRNA and mRNA molecules, catalyzes the formation of peptide bonds between neighboring amino acids, moves along the mRNA in the proper direction, ensuring amino acids are incorporated in the proper order, and terminates the process by dissociating and causing all the components to separate. Prokaryotic and eukaryotic ribosomes are similar but not identical. In both cases, the ribosome has 2 subunits. In prokaryotes, those units are called the 50S and 30S. In eukaryotes, they are the 60S and 40S. The S stands for an old unit of measure called a Svedberg. Basically, bigger numbers mean a larger size. But it's something of a frustrating unit since it's not strictly additive. The 50S and 30S subunits combine to form a 70S ribosome, while the 60S and 40S subunits form an 80S ribosome. For both eukaryotes and prokaryotes, the large subunit has multiple rRNAs and many proteins, while the small subunit has fewer proteins and only a single rRNA, though the number and size of the rRNAs and proteins vary between prokaryotes and eukaryotes. Ribosomes are abundant throughout the cell. The average E. coli cell has about 10,000 of them. In E. coli, there is only one copy of each gene encoding a ribosomal protein, but many copies of the genes that encode rRNAs. Think about why that might be. Let's look at the structure and function of the ribosomes. The small subunit binds the mRNA, while the large subunit possesses the peptidyl transferase activity, catalyzing the formation of peptide bonds between amino acids. When the large and small subunits are together, they form 3 tRNA binding pockets: the aminoacyl, or A site, the peptidyl, or P site, and the exit, or E site. Now we'll walk through the steps of translation, starting with initiation in prokaryotes. Prokaryotic mRNAs possess a site called the Shine-Dargarno sequence a few nucleotides upstream of the start codon. The rRNA in the small ribosomal subunit base pairs with the Shine-Dargarno sequence, which positions the start codon in the P site. The tRNA that pairs with the start codon is not a normal methionine, but a modified amino acid called N-formylmethionine, or FMET. Then the large subunit of the ribosome binds, and the process is ready to begin. There are also proteins called initiation factors that play transient roles in helping this all to occur, but we won't worry about them for this course. This slide gives you a better view of the base pairing I mentioned earlier between the rRNA in the small subunit and the Shine-Dargarno sequence in the mRNA. This pairing positions the start codon in just the right spot to initiate translation. In eukaryotes, initiation is a little different. There's no Shine-Dargarno sequence on eukaryotic mRNA. Instead, the small ribosomal subunit binds the mRNA's 5' methylated cap, though this doesn't position the start codon in the right place. So the ribosome scans the mRNA until the AUG codon is lined up with the proper pocket. Then the first tRNA pairs with the start codon. And in eukaryotes, it's just a regular methionine tRNA and not FMET. Next, we have elongation. The start codon and methionine tRNA are in the P site. Say the next 3 bases are UUU, a phenylalanine codon. This codon is positioned in the A site, so a phenylalanine tRNA enters the A site, and its anticodon pairs with the codon. Now, the peptidyl transferase helps form the peptide bond between the methionine and phenylalanine. The newly formed dipeptide is attached to the second tRNA, and the ribosome shifts so that the first tRNA is in the E site. It's now uncharged and leaves the ribosome. The second tRNA has been advanced to the P site, and the A site is free and ready for a new tRNA to pair with the third codon. That new tRNA enters the A site, and the whole process repeats, extending the polypeptide chain by one amino acid each time. Proteins called elongation factors help to move the ribosome along the mRNA and usher the tRNAs into the A site, but they've been left out of the figure for simplicity's sake. Energy for this process is provided by GTP, which functions like ATP, the common energy source we've discussed previously. Here's a molecular view of peptide bond formation. The peptidyltransferase breaks the bond between the amino acid's carboxyl group and the hydroxyl group of the tRNA in the P site, and it catalyzes the formation of the peptide bond between this carboxyl group and the amino group of the amino acid in the A site. Interestingly, it's not a protein that possesses this peptidyltransferase activity, but rather the large rRNA. Occasionally, RNAs can carry out enzymatic functions. Such RNAs are known as ribozymes. Protein synthesis does not need to proceed one ribosome at a time. Multiple ribosomes can be translating the same mRNA simultaneously. Having many ribosomes along an mRNA is referred to as a polyribosome. In this way, a single mRNA molecule can give rise to many copies of the protein it codes for in a short period of time. Translation ends when the ribosome reaches the stop codon. Elongation is halted as there are no tRNAs that can base pair with the stop codon. Instead, a release factor enters the A site and causes the ribosome, mRNA, and newly made polypeptide to dissociate. The mRNA can continue to be translated by other ribosomes. The ribosome can find a new mRNA to translate, and the released polypeptide can carry out whatever role it exists for. Translation is another process that can be easier to understand seeing it in motion, so I've posted an animation to the lessons page. Translation is not necessarily the last step in creating functional proteins. Many require various types of post-translational modifications. Both prokaryotic and eukaryotic proteins can require this, though eukaryotic modifications can be more elaborate. Parts of a polypeptide may need to be removed. For instance, the F-met at the start of prokaryotic proteins is often cleaved off. Sometimes several proteins are all translated as a single polypeptide chain and need to be cleaved into separate units. This appears to occur most frequently with viral proteins. Sometimes, extra amino acids are present on a protein specifically to suppress its function until the cell is ready for it to act. Zymogens, or proenzymes, are kept in inactive states. But when a portion of the protein called the pro segment is removed, the enzyme is activated. Sometimes the proteins are modified with various chemical groups. Phosphorylation is another common way to have active and inactive states for an enzyme. The non-phosphorylated version could be the inactive state, and the phosphorylated version the active one, or vice versa. Another type of modification is glycosylation, adding sugars to proteins. This can have many functions, which you'll learn more about in cell biology. Lipidation is modification with fatty acids. This can help localize proteins to cell or organelle membranes. Ubiquitination is the attachment of another small polypeptide, called ubiquitin, to the protein. This can serve several roles, but it is most commonly used to indicate that the protein is no longer needed by the cell and can be degraded. Let's close out this lecture by looking at how transcription and translation differ between prokaryotes and eukaryotes. A lot of this we've already gone over, but there are a couple new points to bring up. The biggest difference to remember is that prokaryotes lack a nucleus. Because of this, transcription and translation can occur in the same place. Prokaryotic cells take advantage of this fact by coupling transcription and translation together. Ribosomes are translating the early parts of the mRNA, while the RNA polymerase is still transcribing the later portions. This will be important for certain processes we'll talk about later. In eukaryotes, transcription occurs, then the transcript has to be modified, then transported to the cytoplasm, and then translated. So in this case, transcription and translation cannot occur simultaneously. This slide highlights those points again. I thought the text reminder would be nice to have, but I liked the visuals on the previous slide better. Also recall that eukaryotic genes normally have introns that need to be spliced out, necessitating the extra processing step. Now we'll look specifically at transcription. Prokaryotes have just a single RNA polymerase that transcribes all RNAs, but eukaryotes have several, each focusing on a different type of RNA. Eukaryotic polymerases tend to be more complex, with a greater number of subunits. Prokaryotic polymerases have a sigma factor subunit that binds to promoters to initiate transcription, whereas eukaryotic RNA polymerases interact with transcription factors that recruit them to certain promoters. In prokaryotes, the primary transcript is the same as the final mRNA. There is no further processing. But eukaryotes require several steps of post-transcriptional processing. A 5' methylated cap is added, as well as a 3' poly A tail. And again, splicing is required to remove introns. This brings the additional feature of alternative splicing, allowing a single gene to code for multiple polypeptides. And finally, we have the key differences in translation. In prokaryotes, the initiator tRNA carries fMet, while eukaryotes just use normal methionine. The small ribosomal subunit starts by binding the Shine-Dalgarno sequence in prokaryotes, while in eukaryotes it recognizes the 5' methylated cap. Pairing with the Shine-Dalgarno sequence puts the start codon in the proper position to start translation. However, the eukaryotic ribosomes that recognize the 5' caps must scan the mRNA to find the first start codon. One thing I hadn't mentioned is that prokaryotic mRNAs can have multiple Shine-Dalgarno sequences and thus multiple start sites. This means that several genes can be on a single mRNA transcript. We'll come back to this in a later lecture. In eukaryotes, translation always begins at the first start codon past the 5' cap, so a given mRNA will only ever have a single gene on it. Okay, we're through transcription and translation. And with that, our third unit comes to an end. Next time, we start on the fourth unit with cytogenetics.